So
Because it was recording. Oh, now it's
okay,
recording.
now it's recording again. All right. So I think it would at least be helpful to me if you guys could quickly go around the room and say who you are and what you're working on, and then we can get started. Do it. Start with you.
Okay. I'm Mitchell Lewis. I'm a second-year PhD student in Dr. Tara Dean's lab. I'm studying synthetic biology and more specifically trying to put genetically engineered circuits into platelets to use the platelets as delivery vehicles for the products of the genetic circuits.
Very nice to meet you.
Nice to meet you too.
I'm Nathan DeSisti, and I'm an investigator at the NIA.
Okay.
And my lab is focused on, well, we work on aging and proteomics, and very interested in senescence as a hallmark of aging.
I'm Joanna Bruce. I'm a postdoc in the laboratory of Birgit Chini at the Buck Institute for Research on Aging. And we use mass spectrometry-based proteomics in order to identify biomarkers of aging and age-related diseases.
Thank you.
I'm Carmela Haynes. I'm an assistant professor of biomedical engineering at Emory University, joint with Georgia Tech. So our lab basically does epigenetic engineering, right? So we look at chromatin, proteins that, that regulate gene expression in cells. And then we sort of devise really clever ways to make fusion proteins and manipulate the system using rational design.
Very cool. And I think we have a couple of people on Zoom. I see Ellen. Would you like to unmute and give us a very quick introduction?
I'm just sitting in to see what is going on in the world of synthetic biology. I guess in my lab, we're studying UPR and unfolded protein response. So a lot of our research in the future, I hope, will be on aging.
Great. Excellent. And Faye? We'll give Faye a second. If not, could be AFK.
Push the participants button, so yeah. That's everywhere.
I think that's everyone. Ryan's the other moderator. And so I think we can get started, and we have a very broad and nebulous agenda. So yeah, hopefully for the last hour, you guys were talking about potential opportunities and pie in the sky approaches that you would be interested in. And then now we're supposed to talk about potential connections and perhaps continue the conversation on pie in the sky concepts, but how do we bring synthetic biology and aging together? To me, the most logical approach seems to be to have a question in aging biology that requires answers using synthetic biology approaches. But if you have ideas how it works the other way, I would be delighted to hear your thoughts. And so I don't know who wants to get started. Does anyone want to volunteer?
Sure. So now we are looking for potential connections. Potential connections. Okay. Well, I can say several of us were in the first session together, and one of the things we discussed was modulating like certain hallmarks of aging with synthetic biology tools. So it would be interesting to think of ways to do that, and building, one of the things we lose during aging, that's still sticking in my mind from the last session, is that, it's basically loss of homeostasis, right? So you have a perturbation and then you need to go back to homeostasis. So building in these mechanisms, these resilience mechanisms that allow us to the cell to return homeostasis, either in, improving the existing ones or building in new redundant ones. It would be maybe something interesting to try.
So one question that I think still really resonates with me that came up during our session was the as far as I know, unanswered question of whether a cell's age, right? So these can affect the behavior, right, the function of a synthetic circuit in a cell. There's a lot of the models that we rely on pretty heavily. So in order to get your paper into like a big synthetic bio paper or whatever journal, if the reviewer asks you to test in different contexts, guess what we do? Okay, hex cell, HeLa cell versus Jerkat. And there's no sort of dimension of age because we just simply, we're not, I get, as far as I know, most of us in the community just don't work with that as a model. We see it as more of a specific research area than an important, sort of cellular context that we need to really convince ourselves that our tools will work. But if you think about it, I mean, so it's like all those different cell lines we use to take into account different cell type, but just the aspect of, an aged, skin cell versus a younger skin cell, we're not systematically testing any of these things that we build. So we don't know if like differences in the metabolism and protein, levels in those as cells age, if those would have any sort of feedback or impact on our systems. And I think that's hugely important for translation. If we're going to propose to like move any of these circuits into a recipient, had we been modeling on the wrong age grade the entire time.
Like the transferability.
Yeah.
Of the system.
Right.
So I think, and this is an excellent point because it ties into what we talked about in this room during the last hour. And we spent a fairly significant amount of time trying to decide what a senescent cell is. Now, I'll give you a preview. We didn't, because the SEMnet consortium also spent the day trying to decide what that is and came to a similar conclusion. But to your point, and I think the point is critical, and maybe we can turn to the aging biology part to explore this, is there do need to be functional definitions for the phenotypes that we're trying to either study or rectify, either cellular phenotypes or homeostasis phenotypes and definitions. And so if you could maybe talk a little bit more about either what we need to do to even come up with those definitions, or can synthetic biology approaches help us get there? I know this is a difficult question. I think if we answer it definitively, we can all walk out of here with a Nobel Prize. But if we can at least get started.
Well, so one of the so in the previous session, we were also kind of making a bullet list of some of the, I guess, characteristic aspects of the synthetic biology field and how they're related or they could be helpful, right, to aging research. So to your question is, so this matter of defining a senescent cell, and to me, the aspect of time, right? When does it become senescent? Now you're sort of including this aspect of time, right? So you're having to consider dynamics. And it's so that's one of the strengths or one of the characteristic aspects of synthetic biology is that our aim, we value building things that can respond to dynamic changes. So it's all about, there's the time aspect that we pay a lot of attention to. We're not only interested in just sort of like single states at like single time points, although a lot of us have the bad habit of reporting endpoints. Because the time trajectory stuff is just a lot of information. It's hard to put in paper, but there's a lot of like sort of that's kind of like a key activity in the practice of synthetic biology, is building things where we can actually track trajectories in real time in living cells. So we basically prioritize that a lot. And then so given that, there's emphasis in building tools that will not, mess up the cell too much, but will allow you to observe more easily some things that you would otherwise have to kill the cells in order to measure. I think having a real-time reporter, right, for as a cell enters senescence would actually give you that threshold. Okay, how much time, and not even, maybe not just a matter of time, but what other sort of like metabolites or other cues in the cells once they hit a certain concentration, right? So that's another sort of a unique capability of synthetic biology, is that we're pretty good at building genetic switches that will only flip when you reach a threshold input. So if the question is how much of molecule blah builds up in a cell before it hits senescence, we can actually measure that in real time. So yeah, I think that sort of the ability to build, because we integrate, control theory, right, and control into our genetic systems, then I think by its nature provides a nice, at least the potential for tools that can actually help to define, right, what that actual aging trajectory is. When does a cell enter senescence? When do these key events happen? We could potentially track that.
Would we be able to do this in a living animal, or is this largely confined to in vitro systems for the time being?
So the answer to that is that it's, we can get you a cell that produces an output.
Okay.
So if you can come up with a machine that could track the cell inside the animal, then we're golden, then we're good to go. So I think that was one of the points that came up, is that, okay, well, folks, engineers have done a pretty good job with, continuing to increase the resolution, in space and time, right? So collecting data points over time, and then also getting, sort of higher resolution images out of an animal. Things are sort of improving along those two aspects. So I think, based on what I've heard, we may be at the point, or at least we're quickly approaching it, to where, yes, the synthetic biologist could build a cell reporter system that would give you enough signal that could be picked up by, some imager, some fancy imager. I think there has to be, sort of like the two sides have to approach each other. But that to me is the challenge, is that, because we can engineer at the single cell level, it's just a matter of having the machine or the instrument being able to follow single cells. Or penetrate wherever you need to get, inside the animal to do the imaging. And then there's been some creative thinking around different types of outputs, but we're very limited. So it's either going to be visible signal. Anything you have to talk about to a physicist and the rest of the stuff. But I don't know, like, what other kind of physical outputs you could pick up.
I've heard of acoustic outputs, although I don't know
The
how.
cell's actually screaming.
Yes, that's interesting.
Yeah. But in terms of inputs,
Yeah.
there's a lot more flexibility on the input side. Like, I've seen people present genetic circuits that can respond to magnetic fields of a certain strength. It gets really crazy. Certain wavelengths of light is actually very well developed. And then all kind of chemical compounds, so sort of physical ligands and whatnot. But in terms of the outputs, there's not as much creativity on that side.
So, and what about sensing multiple factors? Because we were discussing that aging is a combination of hallmarks and
Yes, yeah, I think
so is it?
So it could be an and
Yes.
situation.
Like-
There's so a lot of activity around signal integration. So that's kind of like a whole pillar of synthetic biology and applying machine engineering principles. So yeah, your system is basically just going to be really dumb. Like when I teach my students, I tell them, okay, it's the difference between a light switch that will only flip on and switch back off again on its own, versus a light switch that can tell who you are when you walk into the room and how many people are there. And then come on. So you can, genetic systems actually have some inherent properties to where you can actually build multi-signal integration, and it will only switch to a certain state. You can even get so if you apply Boolean logic, for instance, like the and, not, nor, all of that stuff, you can build genetic circuits that will take I've seen an example where there were up to 5 inputs, right? To where it would only respond. Yeah, Jim Collins is really good at this stuff. And it was as fancy as, okay, 2 things are present and the other 3 are absent. And if you have any other combination of those inputs, it will not respond. So there's definitely work. I think it's possible, right, to integrate multiple cues from the cell to make it more specific and even more sophisticated. Yeah.
Yeah, so I think that's really cool. And it's, I think, interesting to, I guess, just thinking about how these things can be used in a collaboration, right? Would these be, these are things I'm thinking of can it be used in a way, is it more to probe the fundamental biology? So again, so now we have some logic that integrates one or more aging signals, like
Yeah.
maybe something very important for senescence. I
Yeah.
mean, that would be interesting too, just to even see what threshold you have to reach,
Yeah.
of course. It might be hard though, because there are multiple candidate proteins or whatever that might
That's true.
be part of that. If there's a way to screen multiples or in a more high throughput way, that'd be cool, but integrating multiple aging signals is also something that,
Yeah.
like you're saying, is very interesting. Yeah, and I know this, so we had the senescence conversation earlier, and they're dynamic. Joanna brought that up in the last, right, they're so dynamic
Yeah.
and heterogeneous, so it's really tough to do some of these things, but
Yeah.
hitting down some very key features like that, like that,
Yeah.
what really determines, where is the point of no return for the cell to choose to become senescent, but,
So maybe
I
there's
also-
some, like the molecular geneticists would know, because I'm not very familiar with what work goes on, but one experiment that comes to mind, right, for answering this question. So to me, that's a question of causality, right? So like, okay, so
Yes.
you observe this feature showing up in the so-called senescent cell. Is it a driver? So the cancer people love to use this, is it a driver or passenger, right? So it's like that feature, right, that phenotype. Is that a consequence of something else happening, or did it happen and then push the cell to senescence?
yeah.
So I think a good sort of, I wouldn't say it's a synthetic biology experiment, but we're keen on creating an artificial situation to determine causality, right? So in epigenetics, this is an important concept. It's like, okay, this histone acetylation. Yeah, we see it show up whenever cells are active, but did it actually cause the activation? So you can use something like epigenome editing to artificially put the acetylation there, and then you see the gene expression go up, you're like, aha, this is at the beginning of that chain. So I think, yeah, it sounds to me like maybe there's more work that needs to be done in sort of taking these things that are observed in senescent cells and then somehow artificially creating one or two or three things and seeing if you can artificially produce senescence that way. Because I got a quick lesson on the techniques that are used to induce senescence, and I was maybe not totally surprised, but I was like, okay, so irradiation and basically poisoning the cell and
Yes.
treating it with chemo.
What
It's
about
like-
space? Cells, because.
Oh, isolation. Do
Isolation.
they go into senescence on their own if they're isolated for too long?
Some will.
Yeah.
We've got some OP9 mouse bone marrow
Yeah.
stromal cells.
Or with OP9s, yeah.
They're pretty common, but they get sad if they're lonely. So
Yeah.
Oh, that's sad.
yeah.
Well,
Yeah.
yeah, they also count as a big, well, so, do those also have like the doubling phenotype with senescence, like the senescent cells tend to get bigger?
We usually toss them once they get senescence.
Okay.
Well, that's the
Yeah.
difficulty too. There's so many different inducers and,
Yeah.
a lot of them happen to be DNA damage
Yeah.
inducers, but there's other things as well. Even mitochondrial dysfunction might actually go through that pathway, or oncogene activation, telomere shortening. And even like there's a signaling pathway like paracrine senescence. So
Yeah.
senescent cells the SAS can transmit senescence to other cells. So there's obviously a
Oh.
signaling pathway as well. So, it's kind of difficult to study because we know senescent cells, there are so-called good ones and bad ones. So even if we know exactly what drives the sulfate, which is extraordinarily important, do we know that the different types of senescent cells, do they all become senescent in the same mechanism?
Oh,
And, and if they all use the same mechanism, how do we then, when we come up with interventions, distinguish the good and the bad ones?
right,
When we want to
right.
make these interventions,
No,
and before we talk too much about it, I just want to say, I think senescence is a really good area of collaboration because we have so much data now, and I think things, tangible stuff, data we can use probably,
Mhm.
like omics. For me, of course, I like at the surfaceome, I always feel like there's something we can use there. But also we should keep in mind, we're talking about aging, and Joanna pointed out that senescence is just one of the hallmarks. And it's not the most important hallmark probably, or if we cured senescence, we're not going to cure aging. We're going to have a small impact probably on our lifespan. So we have to tackle multiple things
Mhm.
together to, approach aging. It'll be interesting to think about multiple things. I just want to say that too.
Oh, yeah.
Yes.
Is the inflammation, the the discussion about inflammation that came up, or at least the introduction of it during this morning session really made my ears perk up because it seemed that you really have this aging as, I guess sort of chronic or the inability to heal.
Oh, yeah.
Chronic inflammation.
Yeah.
And then, well, it made my ears pick up because as a chromatin person, right, it's the innate immune signaling pathway heavily regulated by a certain class of chromatin known as the polycomb repressive system.
Yep.
Every time we go messing around with polycomb proteins and cancer cells, so we use engineered proteins to do it. Other labs use inhibitors of chromatin modifying enzymes to do it. And it's the same almost nearly the same set of genes always gets upregulated when you go messing with polycomb chromatin. So yeah, from there, it's like, yeah, so I've kind of like kept that on the radar because it seems to be cells for some reason are using epigenetic modification as a really, I guess, sort of a powerful regulatory mechanism for immune signaling.
Mm.
And I don't know, I mean, so that's just at the genetic level, but then all of these, sort of like physiology, like cell circulating, that's kind of beyond, what I'm looking at, but yeah.
Go ahead, Jenna.
Oh, yeah, I wanted to say that's true that we saw like some common things like, for instance, looking at the tabs, the secretory phenotype, like whatever the cell type, the tissue, or the inducer, you will find these strong soluble factors that are like released by the cell. So yeah, there are like for sure some like commonalities
Yeah.
among aging and maybe some like tissue specificity then or like other external factors that also influence. Yeah.
I wanted to make two comments in response to this. Has been a really good discussion. I think the thing that distinguishes good senescent cells from bad senescent cells is often the same one that distinguishes good inflammation from bad inflammation, which is severity and quantity.
Right.
Maybe 1 in
Like
100
duration.
senescent cells is good and it can be
Yeah.
safely removed. 15 in 100 is too much, leads to tissue damage. And
Yeah.
same thing, if you completely suppress inflammation, you also suppress the natural healing response and you die.
Right.
You have too much inflammation, that's a problem of its own. And that is one of the contributing factors to tissue damage, especially with age and with other conditions, is uncontrolled inflammation. So I wanted to follow up on a point that Nathan made, that senescence is an area of collaboration, but not necessarily the only aspect of aging. So what are some other aspects of aging that we should talk about or think about?
I may speak to I'll circle back to what you very originally were talking about, was the difference between a young cell and an old cell.
Mhm.
And if you take a cell out of that environment, does it know that it's old? Is it still old? Can you tell them apart?
Right.
But there's one way to tell them apart is through the epigenetic clock. So, we are very limited in how we can tell the age of a cell, but there are certain things that it has a memory of to know it's old. Those kind of things would be interesting to examine. What are these factors intrinsic to the cell that are part of aging versus environmental or extrinsic things, which we had also talked about a lot in the last session. And if we can, there's some way to interrogate those intrinsic mechanisms themselves, because at least this is in an isolated cell system. Maybe it would be a little bit easier to work with, or something that Carmela, you're like in the realm of what you're used to thinking more about.
Right.
So an intriguing question actually comes to mind, because often when we talk about age, we think of the actual passage of time, but that's not really what we mean by cell age. I think what we mean is how close is the cell to dysfunction, and try to identify whether through telomere length or methylation abundance or any other number of epigenetic clock approaches, how close it is to becoming a bad cell. And in theory, whether it's been alive for 5 hours or 5 million years is irrelevant in that question.
Yeah.
Biologically versus chronological.
I think that point was kind of, I think, driven home by this, I think around the beginning of some popular media, that a team had managed to get a fetus right, from IPSC to like some blastocyst, some really critical
I
stage
think it
of
was stage 5
development.
or 7, yeah, it's
Yeah,
fairly recent.
And so it completely
publication.
violated time. And then so
Wow.
they're like, okay, well, now is, are the regulators gonna now have to change their definition of, when you can stop human
Oh,
development,
yeah.
right? Because
Yeah.
that's part of regulation is that
Yeah.
it can't be older than blah days. They're like, oh, but we can actually get it to this biological stage before that. So
mm,
now, yeah.
I haven't heard of that.
Oh, wait, or did they do it in?
No, it was a mouse. It was a mouse.
Okay, they
It
did
was-
it in,
yeah,
right, they did it in a mouse, right?
But-
Damn.
Yeah, so that kind of
But
complicates
I think- don't
But anyway,
quote me
sorry
on
to
this
that
because
point.
it's been a while since I read the paper, but I think they could get the embryo to like a day 7 stage, but
Yeah.
it took a lot less than 7
Yeah.
days.
Yeah.
Hmm.
Yeah.
But it had the genetic markers of what you would, and also the
Right.
morphological markers of what you would
Yeah.
expect. I also don't think it was actually a viable embryo,
Oh,
so left
okay,
to its
okay.
own devices, it would not actually develop into a
Right.
mouse. They could
Oh.
recapitulate certain developmental milestones that you would expect to see in an embryo. But it was, and I think they made it clear for ethical considerations, if nothing else, that there's no way this would lead to a viable animal if it
Right.
were allowed to
okay.
live.
Mm.
But no, that's also, that's an interesting connection to it. But so anyway, Nathan, didn't mean to derail, your thought, or, Michelle, if you have any ideas on what things other than senescence we can, turn our attention to in the realm of aging.
I'm kind of curious about if contact inhibition plays a role in aging. I know that it's often cited with like naked mole rats and their resistance to cancer from their hypersensitivity to contact inhibition. And contact inhibition can be kind of like a temporary senescence or like a pause in the cell cycle. And I'm wondering if that is discussed in aging at all, or
So is that like quiescence versus senescence?
Kind of,
Yeah.
yeah.
yeah, I guess temporary senescence
Yeah,
is quiescence.
Quiescence.
That's a
Getting my
good,
vocabulary.
yeah, that's a very interesting thing to also examine. I think, and that also is related to the contact inhibition or extracellular matrix, like these signals can also be dominant. So even if you have like a cell that's precancerous or something, but if it's in that environment, it might override it and create it,
Hmm.
keep it as a healthy cell or something. So these kind of engineering also could be interesting approaches. There's a paper that just came out from the naked mole rat thing. Because a while ago, high molecular weight hyaluronan was identified as like the thing that makes the mole rats part of their longevity and their complete immunity to cancer or whatever. But now they made a mouse that, in a paper that just came out like in the last few weeks, that overexpresses that gene from the naked mole rat in the mouse, and then they had, longer health span and lifespan. So they kind of exported the aging the anti-aging mechanism a little bit from the mole rat into the mouse. Yeah.
I always kind of thought it was kind of. What journal was it in?
I don't know the journal actually. I just know the study. But you could Google it probably. Hyaluronan mouse lifespan or something.
Increased hyaluronan by naked mole rat has to improve health span in mice.
Nature.
Do you know how much?
It's a small this one.
Okay.
It was like 6% or something.
Okay.
So this was very modest. Yeah. They had more of an impact on the health span, but not really. I think on the lifespan.
Small but statistically significant
Yeah.
increase. Jenna, what about you? What do you think?
I think it's a bit like outside of what we discussed, but like how like synthetic biology could help to have like more personalized response to like aging or like really early onset age-related diseases. How can we have like tweak the tools to be really specific to one individual and one condition?
Personalized medicine.
Yes, yeah, exactly.
Yes, I think that, one area in synthetic biology is, we're constantly looking for different, genetic components and signaling proteins that just naturally seem to be specific for, you know, some key molecule or whatnot. So that's, yeah, it's a huge, but it's rapidly expanding, right? Because, you know, thanks to evolutionary biologists, right, we have some clues about what cells actually care about. Okay, is this conserved across species? Okay, one of these things act different. So yeah, I think there's, you know, a lot of promise. It's just that I think the major bottleneck is the interest in the funding, right? I could go chasing down, you know, some promoter that's specific for an aged liver versus a younger one, or you can sort of find enhancers and promoters and all sorts of things that you could then interface with this sort of like more generic circuit that then produces some output.
Yeah.
So basically that'd be our approach, is to look for something that responds, that naturally responds in a specific context. And then our hope is that after having done this for long enough, we start to learn the design rules. It's like, okay, well, other than just relying on there being something there,
Yeah.
we don't really understand why that combination of ATCs and Gs does this. You do it enough and you find enough different elements, and then eventually the idea is that you get to a design rule. Oh, this many A's in a row followed by N's followed by this gives you this age-specific promoter, right? Or what have you.
Yeah.
So there's in practice, we have a strategy for how we would get to that point.
Yeah. Okay.
Yeah.
I'm gonna ask you a really hard question
Okay.
since you opened the door. But speaking of evolutionary biology, what we tend to find is that the elements of DNA that are the most conserved, especially across multiple species and unrelated families, are the ones that are involved in a large number of different pathways, because that's a great way to
Oh,
get
right.
conserved.
Right.
And so they also tend to be some of the worst targets for treating a specific condition, because we can cure diabetes by shutting down metabolism. I think we
Right.
can do that. Whether
Yeah.
the cure is gonna be
Right.
worse
Right.
than the disease
Right.
is a different question. But I'm actually not trying to be facetious. I think this is
Right.
this is ultimately where we want to be going. And the question that I struggle with conceptually is, do we have ways of deconvoluting that, either for therapeutic purposes or coming at it so that you take a pathway that is very general, but limited to a specific context reliably?
That's a great question. Can you context sensing?
Yeah.
Can you
Right.
build context sensing into it?
Yeah. Specific. Yeah. So I
Seems
think,
like
well,
there-
part of the answer is signal integration, right? So it's, you can kind of get around that, right? If you have a, right, so if you're trying to make a, basically what is in Boolean logic a buffer, if this is present, then do this, right?
Uh-huh.
Very bad, right? Probably
Very-
not very robust, not very specific. The more complex you make your inputs,
Mhm.
then that's one way to, and it doesn't, I don't think that you know, the maximum, like, to be useful, maybe 3. I mean, it does not like you have to have like 20 different inputs to like make, because then once you get past a certain point, the thing is just useless and it's probably not gonna be very robust.
It'll never trigger.
Right.
You'll never get the conditions.
Yeah, so like if you could have like, I don't know, even just 2 or 3 inputs,
Uh-huh.
then you could, yeah, make it more specific, but not have it, right, I guess, I don't know, that's something I can think of off the top of my head. But no, you're right, that is a really important problem. It's, you know, yeah, the more, I guess, sort of functional and well-used this, you know, particular biological component, then you have to worry about specificity because you lose that. Yeah. But then it's like, okay, so here's an even harder question, right? So it's like almost produces this chicken and egg scenario. It's like, okay, do we need to learn more about aging in order to engineer it? And so, but then we need engineering tools to better understand it.
Yeah, no, that's a very fair question, because I personally, if you ask me to define what aging is, other than the thing that causes you to eventually die, I would be hard pressed to come up with a more definitive statement. So I think, at least in the short term, thinking of it in the context of specific problems is probably what we have. We love calling them use cases. I mean, like, okay, but I think that's and I realize it's a little bit of a cop-out, but the truth of the matter is, I don't have a good answer of what aging is, and so now, granted, I'm also not an aging biologist, so don't listen to me.
Right.
But it would be hard to reverse or prevent something if we can't exactly define it in the first place.
Yeah,
Yeah, that's why we kind of go back to the hallmarks of aging, because these are the things we know may be causatively linked to aging, at least they drive some of aging. And there's certainly hallmarks, they're certainly there at the right time in the right place to be part of aging. So those are things we can already begin to intervene, and I actually don't think we fully really fully understand the extent to which these hallmarks are driving aging. And we talked about in the previous session is the order, the causative or the order of events too. Like what's the causative driver? They all affect each other. What is the sequence of events among the hallmarks? Those would be interesting questions to ask.
that's kind of one kind of maybe annoying debate, kind of big question. So I just have this picture in my head where it's like, okay, well, now aging, yeah, I think it's important to be careful because there are gains that you make, right? So it's like, okay, is a completely naive, there are cases where a completely naive state in a biological system is bad.
Mhm.
You're either gonna survive some insult or you die, right? So then if you survive that insult, you are now trained. So immune system is a classic example. So it's like, I think it's important to think about how and when, and I think the aging people probably have this figured out, but how and when resets, right? So this whole concept of a reset, I think that's one application that folks who are, researching aging are very interested in. But I think it's important to think about cases where a reset could actually put the system at a disadvantage. So you don't want to reset too far back. I mean, how young do you want your system to be? So maybe there's like this sort of like window that you want. So there's, the degeneration side, but then there's also, I think maybe I'm thinking that there's a state where the system is so naive that could be a detriment.
Yes, definitely. I think the epigenetic reprogramming is probably a really good example.
Oh, so actually, so so one thing that I keep thinking of, and I hadn't had a chance to bring this up yet, every time someone talks about epigenetic reset and stem cell cancer,
Exactly.
right? So
That's
that's one of the
why-
tricks
that's
that
why-
cancer uses, right?
Yeah.
Is to restore its proliferative potential. And it's like, okay, we probably don't always want that.
Well, if you want proliferating cells,
Well, yeah.
cancer is fantastic.
Right. But then your point is balance, right? You have to be careful, pushing things too far in one direction. You want to be able to control proliferation in a way, but you want to still be able to achieve homeostasis, right? So that's just one thing that bugs me every time I read like one of these, reversing the cellular clock papers is like, oh, we reestablish iPSCs and like, okay, well, that's what cancer does. And I still haven't read any kind of very satisfying review that really sort of tackles or a viewpoint or perspective.
Oh, yeah.
This seems
It's
like
very
the
well known
elephant
in the
room.
field now that, aging biologists know the epigenetic, that's why they say partial reprogramming.
Oh, right, okay,
partial,
right.
Because they don't go all the way back because of teratomas and cancer.
Right.
So, yeah, I mean, it's a little bit of a controversial space, but now a very popular space. And like, there's a big company like Altos Labs who took a bunch of people out of the academia who are really big, hitters in aging, working on this.
Yeah.
Now, I agree. I feel like it's a very, very beginning stages of this field. Like, it's gonna be very hard to reprogram just the right amount.
Yeah.
And in the right cells, so that there's no danger. So I think a lot of researchers will have to kind of play with different approaches or kind of doses and scheduling of these reprogramming factors, right, to cells. Yeah.
Which actually ties.
Could I jump
Okay.
in? Sorry. I just wanted to say, instead of just focusing on reprogramming cells, I think there are also, for blood cells especially, the clonally expanded cells, the ones that have somatic mutations that seem to allow them, to give them an advantage in replicating. Some of those, which are associated with a lot of causes of ages, targeting, just eliminating those that have the advantage of replicating, you wouldn't need to reprogram the cells. You just need to get rid of some of the cells that are produced in excess during aging. And so that, I mean, you wouldn't have the danger of potentially starting cancer if you were to focus on that, or potentially eliminating cells that have lost Y chromosome expression or something like that.
Yeah. Is there a way we could detect that and then essentially address it immediately in vivo? So if, for instance, you lost a chromosome, is that something that we could, using synthetic methods, A, detect, and B, eliminate without destroying the tissue, the surrounding tissue?
Yeah.
I'm just thinking of the most extreme example. I mean, there's sounds like there's some much more fine-tuned changes that would be harder, but
Well, I mean, if I were tasked with that as a, like, I guess, maybe for instance, an undergrad item participant. I don't want, like, a very naive proposal would be to, okay, well, you'd have to engineer a cell, but put a transcriptional repressor on the chromosome that you don't want to lose. And then put like some toxin gene on a different chromosome. The repressor shuts off the toxin gene. So if you lose that chromosome, you turn on the toxin and that cell dies. So you kind of basically put in like a suicide switch. So yeah, I mean, all I have to say is that I think, yeah, making sure that something is present in the cell the initial approach to that would be pretty straightforward.
And I think you could cover a few things with it, but if you try to cover every chromosome with a suicide
Oh, right, no,
switch,
right. Then
you would fairly quickly run
that's
into a
where
problem.
that's why that's an undergrad. Right, yeah, but it would, be very limited in use, but yeah.
But it would
But
be
if
very
you
specific.
came up with a more sophisticated, right, system to account for that, then I think it would be feasible, but.
Thank you, Ellen. Do you have any follow-up based on what we we're just talking about? No. So let's see, it is,
Oh,
we
we
have 15 minutes. I've been trying to take notes.
Okay.
So I think I will probably report out for this session. But this is a good prompt that if we now 45 minutes in have some very specific concepts that we should make sure get included in the report out, then this would be the time.
Well, man, speak. So, I think about, from things that are very practical to me, because of course I think about in terms of, what kind of tools do I have and data do we have is, I think, very interesting. And from things you presented too, Carmela, was the, the cell surface receptor, and targeting those. So first, in terms in senescence, now we have those, so we can come up with systems either to payload delivery, to target those or something that would be nice. And the nice thing about the surfaceome is that, senescent cells are very, very heterogeneous, and maybe we can actually distinguish different populations of senescent cells specifically with that. So it'd be a more targeted type delivery. I think that's kind of a lower hanging fruit for me. Yeah, in terms of the cells, I guess looking, I think a lot in terms of interventions, I guess, too. So, I guess there's that thing in the fundamental biology, which we were also talking about earlier. So like if you took a cell and, just say you, epigenetic aging in an isolated cell, can you build something into the cell where it like resists the changes, the normal epigenetic clock changes? Because if you can directly alter those to see, is it like, does it have some kind of positive role in aging? Like just preventing those, does it keep the cell younger in other ways?
Yeah. And I think it's, with that type of project or experiment, as a chromatin person, I would say that it would be super important to mine the metabolome, right? So it's not, I think when people think of chromatin remodeling, they immediately start thinking of the chromatin remodeling enzymes, and that's great because they're central to that. But you can stop a histone acetyltransferase in its tracks if you deplete acetyl-CoA. That's all it takes. It only has one substrate. So the substrate pool is, I think, just as important as like, the levels of histone acetyltransferase proteins. So in that type of I would, so if I were in a position to sort of track that, I wouldn't know much about how to actually follow the pool of acetyl-CoA. So that's where that would be the bottleneck for, my research lab, for instance. But it's super important to look at because you could be looking at two different cells that have exactly the same level of HAT proteins and they have wildly different levels of acetylation. It's like, why? Well, you have to look at, how much acetyl-CoA is available. Another major one is SAM. SAM affects DNA methylation and methylation of all histones. The set domain containing proteins, the methyltransferases, only use SAM. And then another big one is 2OG, which I think is produced by the mitochondria. And then that is a cofactor for histone deacetylases. So it's like, so the metabolism, so I'd be curious about how fluctuations, because it's like diet, exercise, all of that, but how do fluctuations and I guess the steady state levels of these major epigenetic metabolites, those might be pushing, aging, like maybe if you deplete for too long and then you try to reintroduce it back, maybe that does something to the cell's memory. So I think, as much as of a fan, I'm very protein centric on the chromatin side, but I cannot deny the importance of changes in levels of metabolites. And so having tools to track that, and I guess that would be sort of on the, unless a synthetic biologist has a really good tool for following those in living cells, like our chemistry and metabolomics people would have to come in and help with that.
So before this becomes a surprise, I received some updated marching orders because I might already be doing a report out for one of the sessions. We need a volunteer.
Oh. Okay. Sure.
You have some nice notes.
Yeah, I've got a few. I probably don't have as many as yours.
I have different notes. They're mostly
Okay.
chicken scratch, but we can compare notes.
So, okay, so then I guess just to recap really quickly, I guess to just directly respond to the prompt, right? Some of the things that came up in the discussion in like connections between research questions and approaches. So for instance, one that I wrote down because I said actually probably doesn't say this. So basically determining the molecular drivers or at least things that maintain an aged state, right? So what actually induces it at the molecular level? So besides, the radiation and all of these, insults from the environment, what could you, and I guess that's a setup for, okay, well, you would design an experiment to where you would test causality, right? Change something, manipulate something in a cell and see if you can induce an age state that way. In a way that hadn't been done before.
And it's a little bit tough though, because then we don't have a good readout for how do we know if we're in an age state or not? That's the difficult part, to do this in cells.
Right.
And that's up for like epigenetic clock or, yes.
I do really like the idea of considering the metabolic effects and epigenetic modifications. That's honestly one of the best ways I've heard that connection articulated. Thank you. Jana, reading something?
No, I mean, I was thinking like when you was mentioning earlier, for instance, if you read like the epigenetic alterations, then have like a way to intervene to restore it. And coming back to this like combinatorial effects, maybe like it's more like a blue sky idea, I don't know, but like even have like a combinatorial responses, like maybe intervene on the epigenetic level, but you mentioned like the metabolic level is also important, so maybe also in parallel intervene on the metabolomic level, and maybe like other hallmark of aging on, so like have, I don't know, like multiple input and multiple output tools.
Yeah, that's great.
Let's-
Payload delivery will be an easy one for not only just
That's that one.
for anything, yeah, for any,
Yeah.
I mean, in many contexts, yeah. Targeting.
So, and circuit size. I suppose the more biomarkers that we're trying to look for, the bigger the circuits are gonna be and the bigger issues we'll have to have.
They're kind of trying to control complexity because too few inputs may not be specific enough, but too many inputs essentially requires you to win the
Yeah.
jackpot and may not be a useful circuit.
Yeah, like trying to have a supercomputer versus a laptop.
Right.
Well, there's time and place for both.
Exactly. And a set amount of resources in situations.
Or Carmela, what you had initially described too was interesting, that idea where you can track, like synthesize in a protein and track thresholds for it to enter before it
Oh,
becomes a
right.
bad cell, right? And
Right.
something you can actually even use that to image maybe in vivo. I don't know, maybe that's more complex, but either in vitro or in vivo, both would be interesting ways to probe
Yeah.
basic biology of aging, yeah.
Like a spatial and temporal
Yeah,
type of readout.
yeah.
Ellen, if you're still with us, do you have any thoughts on what you'd like to see included in the report out, if anything?
She's fine. What did she say?
Sorry, I wasn't able to unmute myself.
Oh, it's okay.
I think you guys are doing a great job. I just, yeah, have other stuff going on in the background here.
No worries, no worries. It looks like we're gonna have kicked out in 50 seconds, so just wanted to make sure we give you a chance to talk.
Yeah, well, thank you so much. It was really interesting to listen in.
Thanks for coming.
It was a fun discussion,
Yeah.
you know.
Yeah. No, thank you. This was great.
So what kind of, I mean, your background's not either in synbio or aging, so now what are you working on currently?
So that's a very interesting question.
Yeah.
I'm like a program leader with no portfolio right now, which means I'm
Yeah.
working on a whole bunch of different things. I work on encouraging and enabling